It is 10:31, so we are going to start this training for Chlorophyll A. My name is Paul Steen. I work with the Huron River Watershed Council. That's an organization based in Ann Arbor, but we have been, me and my predecessor, have been involved in MiCorps since the very beginning, in 2004. So the Huron River Watershed Council has a long history of working with lakes and stream groups from across the state, and training and working with data, and we're happy to continue to do so now going into this new decade. So today we're going to be talking about Chlorophyll A for the next hour. Here's the outline of what we're going to talk about. As you take all that in, one thing to mention is that chlorophyll is considered one of our more advanced parameters. And what that means is that you can't register for it until your second year. And the reason for that is because we don't want you to get overwhelmed with so many new procedures all at once. So the first year you come on, you can do the phosphorus, you can do Secchi, you can do score the shore, and you can do the exotic plant watch. But the other parameters are kind of reserved for your second year or beyond after you have mastered the initial protocols. You know, it's not our intention to scare anyone away with learning 8 different protocols all at once. So that's why we take it slow and we ease you into it. And as we go through what it takes to do chlorophyll, you'll see it is a lot more complicated. There's more ways to mess it up. And so that's why we ask volunteers to just take it slow, be patient, work the system, because history has shown us that it works. So with that in mind, we are going to proceed with Chlorophyll A. So first of all, what is Chlorophyll A? Well, as Eric mentioned earlier, it's one of those trophic state indicators, you know, that tells you if your lake is on that, where your lake is on that continuum of oligotrophic to eutrophic. And it correlates nicely with transparency and total phosphorus, and the 3 of those together give you a really good picture of the nutrient status of your lake. And then the dissolved oxygen and temperature, which we'll talk about later, is, like Eric said, this shows the symptoms of that. So chlorophyll, now you probably all remember learning about chlorophyll at some point. Often it comes up in your high school biology classes, and it's that photosynthetic pigment that is in green plants that helps capture sunlight and turn the sunlight into sugars that the plant needs to survive. Now, why chlorophyll A? We say we're monitoring for chlorophyll A. Well, it turns out there's also a chlorophyll B, and there's a chlorophyll C, and there's a chlorophyll D. And because I hate to say I know everything, there could very well be a chlorophyll F through Z for all I know. But there's this wide range of different molecules that all form this family of chlorophylls. Now, chlorophyll A is most likely the one that we are going to be finding in our area of the country. And it's represented in green algae. So we're looking for chlorophyll A because it's the most abundant type in our area. Now, when we talk about chlorophyll A and its presence in algae, our minds and our pictures, if you do a Google search, will immediately go to that shot on the left, and where you see super cloudy, you know, hypereutrophic conditions, algae blooms. And that's maybe what you think about when you think about chlorophyll in algae, but that's only the case for really a few lakes in our state. Most lakes are going to look more like the one on the right. Where we're in a mesotrophic situation, you're not going to see those huge clouds of algae very often. And you may not even consider that there is algae in those lakes, but yes, there is. The algae and the chlorophyll within is forming the base of the food systems in all our lakes, even even oligotrophic lakes, so they will not have much, will still have algae in them. So this will look really familiar if you were just listening to the phosphorus presentation. We have specific times we want you to sample for chlorophyll A, and you see that in the third column. It says sampling dates. Now, this is also broken down by county, so where you are in the state determines your sampling. Everyone samples the same thing for samples 1 through 4, and that is somewhere between the 10th and the 20th for May, June, July, and August. That is when you will be taking one of these samples. The 5th sample is set on the calendar to correspond when you take your summer phosphorus. So the idea is that you can just do it all at the same time. And if you're in the Upper Peninsula, you're looking at doing that sometime at the end of August. But if you're in southern Michigan, you're looking and doing that sometime toward the end of September. So that's because things are already starting to cool off in the UP, and we might already be looking at a lake turnover in sometime in September. So, so the idea is that we're trying to get these samples done before that fall turnover happens. Just like Eric said, we haven't determined exactly when these samples will be turned in. It's very important, and we'll talk about this more later, just to keep everything frozen. You'll have all these chlorophyll filter samples. Just keep them in your freezer till we release all that details about where to turn them in. This is what your chlorophyll data form will look like. We ask you a lot of the same things in every single data form you'll fill out. We'll ask you your lake name, your county, your township, your field ID number. That's that 6-digit number that everyone will have. What makes the chlorophyll data form a little different that you're not going to have on the other ones is, do I have a, nope, I don't have them circled. We have a spot for Secchi depth because that matters. We have a spot for composite sample depth, which we'll talk about. And then at the bottom here, you'll see there's a place to record how much water you are using to filter your sample. So all of those are kind of what makes the chlorophyll data form unique. You'll have one data form that has May and June on it, and then you'll have a second data form that has July, August, September on it. So for chlorophyll, of the 5 samples that you have the opportunity to take May through September, to get your data as a proper approximation of the overall summer months, you need to get samples in at least 4 of those months. So there's a little forgiveness, and you can miss one of those samples if you have to. Getting all 5, of course, is the best to give you that the best overall number for your lake. And just like Eric was talking about earlier, the reason for this is because your lake changes over the course of the summer, and what your lake has in late spring, early summer is going to be different than the way your lake looks in September. The algae is changing over time. So that's why you have to get at least 4 months. You can't just sample the beginning of the year, because then that wouldn't be representative of what's really happening in your lake. Okay, so this is fun for me because usually when I'm in a classroom setting, I'm so far away that I can't properly show people the equipment. But now you see my face right in front of the screen, and I'll be able to show you these things up close so you know what type of equipment we're using in chlorophyll. So we'll start, hopefully my background doesn't mess it up actually. You start with a 60 milliliter syringe. This will come in your chlorophyll packet. This is called the filter holder. And the filter holder unscrews.
You might want to stop screen sharing.
You can't see my picture in other, yeah. I wonder if I can make myself big.
Double tap the mini.
Yep.
What
You're
is
good
different
now.
is that everyone may be able to control their view differently. So if you can't see Paul large and you want to, in the upper right-hand corner, you can switch to speaker view instead of gallery view. And then you will see Paul in a larger screen. Choose speaker view.
Yeah, thanks, Joe. Okay, yep, and now I'm looking straight at myself, which is always a little weird in Zoom. So this is the filter holder. This is called the O-ring, which comes right off. And you can see that there's like a little screen right in there. And that's basically, we're going to be putting a little filter in this thing and shoving the water through that screen. Okay. We also have, this is called the Tigon Tube. And what you do with this is you take your syringe and the tube goes in there. Now the syringe will have these threads in it. The tube does not have threads. That's because these two things were not necessarily designed to work together, but we make it work because all you have to do is put enough force and you can get that in there. You just have to squeeze and push and you end up with that. And that's actually how you're going to draw up your water. Also, when you get your kit, you will get vials that look like this. Well, anyway, they're small plastic vials. Yeah, there you go. And that's where your samples will go into after you filter it on the filter paper. We provide nothing special about these, just tweezers. We provide a pin, and you'll see how those are used in the filtering process to help fold your filter. You will receive a bottle that looks like this, and it's full of magnesium carbonate. And so in the filtering, in the water collection process, you actually need to add some drops of this into your sample. Let's see, these are what the amber bottles are. And this is where you will pour your water samples that you get from your lake. You'll have two of these. And just like with phosphorus, you will have a primary sample and you will have a replicate. Okay, so I just wanted to show you that stuff, but now I am gonna go back to the presentation from current slide. And I have to share screen again. Okay. All right, back to sharing the screen. Now I have some other pictures of things that I didn't cover yet because it's just sometimes it's easier to see on the screen, which, for example, this is a really important one. This is what your filters will look like when they come to you.
Paul, I don't see your slide.
Okay. Thank you. Good?
Thank you, yes.
Looks good.
Yep. Here, let's do that. Okay. So your filters will come in a baggie. And there's going to be a mix of things in here. There's two things. The actual filter that you will be using is white and it's opaque and it's almost a little glossy. That is what the filter is. Now, The filters also come with packaging that is blue and is somewhat translucent. That is junk. And the reason we just throw it all in here is because the filters can kind of get stuck on each other. So this packaging helps prevent that from happening. But when you're actually at the point of using your filters, you want to throw away the blue circles. This will be a problem because some people will try to filter the sample through the packaging and it won't work, and then we'll ultimately have to reject your sample. So just think to yourself when you're doing this, blue equals bad. Don't use the blue circles, use the white circles. Now this confuses people because when you look at the filter is just going to look like, to you it might just look like a piece of plastic, like it won't feel like a filter. And the blue thing will actually feel more like a filter to you because you'll be able to see through it. But anyway, if you remember blue equals bad, that is the best way to not mess up this step. Okay, then you will also get a sampling device called the Integrated Depth Sampler. And it looks like this. This is of homemade construction. Jean and her husband Jim make these for the whole group. This device is a large amber bottle in a juice can. All right. And what it functionally does is allows you to take water samples over a wide range of depths because it has these little tubes coming off the top. So the water is going to enter this sampler slowly over time as you pull it up through the water column. It's fully disassemblable, which you should do between every time you make a sample. You should take this thing apart because if you just leave it all together, eventually it will rust out. So you want to make sure that you take it apart and dry it. This will have weights at the bottom of it. And the weights make sure that it falls nicely down to the bottom of our sampling depth. And those weights are not glued in or kept in here in any way other than just that they're under the amber bottle. So you have to be careful when you use this thing that you don't actually accidentally dump your weights into your lake, which happens because people might forget that they're under there. And then you have to ask for new ones. So don't be that person who dumps your weights into the lake. Keep them safe and set them aside if you're doing any dumping motions over the side of the lake. Now, another thing that you will need to provide for this is a rope. So we will not provide the rope for this. So that's something that you will do, and you want to make it a measured rope. So either with a heavy permanent marker or perhaps electrical tape or other kinds of tape, you can mark off 1 foot increments on your rope. And you'll want to do that far enough that it's approximately double your normal Secchi depth, because that's how far you're going to end up dropping this thing. Now, when you do your registration, you'll also see something that says, do you want, gives you the option to buy a resupply kit. So what's a resupply kit? That's for people who are returning chlorophyll volunteers. You don't need to buy the permanent stuff every year, like the sampler, but there are some consumables that are used up every year. So you need a resupply of those consumables, stuff like the filters and the vials and the magnesium carbonate and so on. So, so when you come back in future years, you will end up buying this resupply kit instead of the bigger kit. For 2021 this year, any returning volunteers who are listening, make sure you throw all your old filters away. Don't reuse your old filters, even if you have established quite a stash of them at this point. And the reason for that is that we just noticed some of the old filters were not working properly. So we have gone through a totally new manufacturer to have new filters that should be doing the job as they should. So get rid of those old ones. You don't need them anymore. Okay, so here's other things that you will provide. And it's very similar to the phosphorus. You need to get out there on your boat, anchor yourself if it's a windy day. You'll need that pencil or Sharpie to write on labels, and then a freezer ice pack, because you always put these things on ice when you are done doing your job and coming back to your house. Okay, so that's the equipment, and I was kind of jumping into a little bit of the procedures to help you understand how the equipment was used, but now we're just going to walk step by step and making and getting the entire sample collected. So the first thing, step one, is getting a depth integrated water sample. So let's talk about what that means. You're out on your boat, you're at your deepest point, just like with the other parameters that you're doing. You're filling out your data sheets. Yep, okay, we'll get to that depth integrated stuff in a second, but to figure that out, you actually need to make a Secchi disk measurement. So this can just be that normal Secchi disk measurement that you would make every other week. You're also, you need to do it as a component of the chlorophyll. So you're doing it anyway, just as a reminder. You're lowering it slowly until it disappears from view. You're raising it up till when it reappears, and you're taking the average. You add the two together and you divide by 2. That's the average. So that's, you're doing that. And then for whatever reason, every time we get a picture of anyone doing Secchi, they're always using their sunglasses, but you're not supposed to use sunglasses. You want to use, because that's going to affect the visibility, of course. So you want to use your naked eyes always to do Secchi depth. But for chlorophyll, we're not doing this at the Secchi depth. We're doing it at twice the Secchi depth. So this is what we're talking about when we're talking about depth integrated samples. And the reason for this is the sun, the light of the sun can penetrate to approximately twice the Secchi depth. So even though you can't see down there because it's beyond your visibility, that doesn't mean the sun isn't getting down there, right? So the sun is penetrating to that depth. That is a depth at which algae is growing. That entire, from that lowest point, twice your Secchi depth, up to the surface, that is where algae is growing. And where algae is growing, that is what we want to sample. So on your data sheet, you will write in the Secchi depth, and then you will double that number and write in your composite sample depth. So you just double the Secchi. What if there's a couple questions here that often come up. What if my lake isn't deep enough to go twice the Secchi depth? That's okay. You just go as far down to the bottom as you can without hitting the bottom. You never want to hit the bottom with your equipment. So you kind of want to know what that depth is prior to lowering things down, because you don't want a plume of sediment coming up if you hit the bottom with your equipment. So in either case, if you have a very clear lake and your Secchi disk is all the way at the bottom, that's the composite sample depth. Or you just basically want to lower that sampler as low as you can without hitting the bottom. Okay, so first thing, after you figure out your Secchi and your integrated depth, is to disassemble your sampler. And this is the point where people lose their weights. So, notice how awfully close he is to the edge there with his sampler. So don't bump your thigh and knock it right off, right? So you may want to be a little more careful than that. But what you're doing is filling up your amber bottle and rinsing it a couple of times with lake water to make sure that you only have lake water in there and nothing else. Okay, then you're going to reassemble your sampler. What you do with the clothespin is mark your line at that point that that is your depth that you're going to drop this thing to. That way you can know exactly how far down it is at any moment, because you can keep your eye on that clothespin. If your integrated sample depth is 30 feet, mark your clothespin at the 30 feet mark on your line, throw all the line overboard, and then you can just release this thing and it will drop. It has these weights in it, so it will drop fast. And you want to stop it when it gets to the clothespin, the depth at which you want it. So you just let it free fall. Then at a slow, steady rate, you bring this sampler back up to the surface. Now this may take some practice, and I can't really tell you exactly what rate to do it, because it depends on your particular piece of equipment and how deep your depth was that you dropped it to. But your overall goal is that you want your bottle somewhere between 2/3 and 3/4 full. So after you do pull it up, take a look, take the rubber stopper and the cap off and look at how much water you actually have in there. If it's completely full, the problem is you may have been going too slow when you were pulling it up and you gave too much time for it to fill up at a lower depth. And the problem with that is that then you don't know if you were really sampling over that whole depth. You may have collected the water entirely on the bottom, and you don't even have any water from the surface that made it into the jar. Okay, so then you just want to dump it out and repeat the process. If it's too empty, you're simply just not going to have enough water to do what we need you to do. So that's why 2/3 to 3/4 full is kind of the sweet spot for how much water you're trying to collect in this process. With your amber bottles in hand, you'll have 2 of those, remember. You are going to fill those up. And then you'll be mostly done on the water. Oh yeah, I was going to mention one thing that you want to do whenever you're dealing with chlorophyll samples is you want to keep things well mixed, but you don't want to shake them like crazy and start breaking apart algal cells and stuff like that. So when you do this step of pouring, you can swirl your bottle, or you can like slowly invert your bottle. You're just doing gentle mixing, not really vigorous mixing. That ensures that the algal cells are fully distributed in your water sample. Then you're going to take your magnesium carbonate and add 5 drops to each bottle. And again, gentle mixing, gentle inverting. And you put that in your cooler on cold storage until you get back to shore. So that's basically all of that is kind of like step 1 of the process. You have now collected your water samples, and now you're going to have to, the more complicated step, filter them. Now this is complicated enough that you really could kind of need to see it in action. So I'm going to attempt to do a video. This actually, I don't have good access to the chat because of the way Zoom works. Could Joe or Eric, can you like even maybe just copy down or screenshot this YouTube link and stick it in chat? That's for your later watching purposes. You can rewatch this video again, and whenever, right before you do your sampling, if you want, as a good reminder of how this works. But for right now, what I'm going to do is share the YouTube directly so that we'll just all watch it together right now.
And I'll also mention that these videos are available directly on the MiCorps website too.
Thank you. Okay, so I'm gonna, oops. I totally moved the screen. It starts at about 5 minutes and 50 seconds is where the filtering point starts. Okay, so here we go. If you can't hear it, turn up your volume a little bit. Even though it takes a couple seconds for
the best place to
sound to
filter a
start.
chlorophyll sample is indoors, by a sink, and out of direct sunlight. Make sure you have all of the equipment listed. Ready to go? First, unscrew and open the filter holder. Use tweezers to pick up and move the filter. Never use your fingers. The filter is a glossy and opaque white circle. Be sure not to use the filter separators, which are translucent and look more like tissue paper. Center the filter on the screen and place the O-ring on top of the filter. Screw the filter holder back together until it is moderately tight and place it to the side for the moment. Grab the syringe and the small piece of clear plastic tubing. You can push the tubing onto the syringe. It doesn't fit perfectly, but it will not slip off if you work at it. Swirl or invert one of the amber bottles to mix. Rinse the syringe by drawing up sample water until the syringe is full, and then empty it. Then fill it up to the 60 milliliter line. With the syringe pointing up, tap it to force air bubbles to the top. Push the syringe plunger until the end of the plunger is lined up with the 50 milliliter mark. Attach the filter holder to the syringe. Like the tubing, it won't snap into place, but you can get it to fit snugly. Slowly push the chlorophyll sample through the filter holder and adjust your pressure to achieve a rapid drop rate. Pushing too hard may rip the filter, so avoid excessive pressure. As you push the plunger, make sure that no water is leaking out the side of the filter holder. If your sample has a lot of algae or other particles in it, it may become too difficult to push the full 50 milliliters of sample through the filter. In these situations, stop pushing and note how much water you were able to filter by reading the side of the syringe. There is a place on the datasheet for you to record this information. After filtering the water, separate the filter holder from the syringe and unscrew it. Using a safety pin and tweezers, fold the filter in quarters with the algae on the inside. The filter will probably stick a bit to the filter holder, and it requires a steady hand and some practice to do this job smoothly. Remember, never touch the filter with your fingers. Put the folded filter into a sample vial and cap it. Place a filled out sample label onto the vial lengthwise so that none of the label is wrapped upon itself. Let's take a look at what a correctly filled out sample label looks like. You need to use a fine-tipped, permanent black marker to fill out the sample labels. In addition to the basic lake information, write CA in the parameter code box and write MgCO3, which stands for magnesium carbonate, into chemicals added. Remember, you added magnesium carbonate in an earlier step. On the label for your second sample, write REP next to the lake name. This is short for replicate. You now have one sample complete. Congratulations! Now you get to do it all over again with your second sample bottle. Once both samples are complete, wrap the vials completely with aluminum foil to keep all light out. This foil wrapping step is critical. Unwrapped samples cannot be accepted for laboratory analysis. Write your lake's name and the sample month on the foil packet. The samples go into a Ziploc bag with your lake name, county, and field ID number. Fold the data sheet, place it into a separate Ziploc bag so it stays dry, and place it in the bag with your samples. Storing your data sheet with the samples is a good way to make sure the paperwork never gets lost. And then everything goes into the freezer until it is time to turn it in.
Okay, so that is the filtering process. Let me just get rid of that there. Sorry that you heard some weird beeping going on. Also, someone managed to draw on the screen, which I think is very impressive. I didn't even know you could do that. And I don't know how to get rid of it either.
Well, what happened there is we do have the annotation turned on. If you go to your, and whoever did it can go in and erase it. But if you want to get rid of it, you have to go up to the sharing menu where it says more, and you can turn off annotation.
Okay.
So it doesn't happen again.
Right. I can hide it, though I cannot seem to delete it myself. Okay.
Yeah.
That's fine. It's just a little scratch. Okay, so just continuing on, some best practices here. Make sure that you're copying your own data forms for your records, and you'll be submitting one of those in with your samples. Keep everything frozen. Those filters, once they go in your freezer, leave them there until it is time to do the turn-in. And finally, there is a database component to this parameter, just like there is for phosphorus and just like there is for Secchi. All that information that you're writing on your data sheet will eventually need to go into our website. And what we do is we take that information and then we match it with our with the result from the lab once the lab processes your samples, and that together lives in the database. So to do your data entry, you need to go to our website, and we want you to get that done by the end of October. Okay. Now it's not unusual to have to reject a chlorophyll A sample for some problem that may crop up during the collection process. So here are some of the main things that you want to avoid as a volunteer. Make sure you're sampling it in the proper time. So for most of your sampling season, that's the 10th of the month through the 20th of the month. And of course, that last month will be different, as we talked about. You will not want to try to filter chlorophyll water through that blue circle. So remember, blue equals bad. That if you do send one of those in, that's an automatic rejection, and we'll send a note to you to remind you not to do it anymore. You need your samples wrapped up in foil. That's because we're keeping them dark. We don't want any chlorophyll growth after the fact of of the filtering and the sampling. So things are wrapped up in foil to keep them nice and dark and away from sunlight, so that what you have on there is what was pulled out of your lake. Now, another reason that samples can get rejected is if perhaps they were incorrectly delivered to us, and often in those cases the sample will thaw, and if the sample comes to us thawed, we can't use it. So make sure you follow our drop-off and procedures. If you can't make those drop-off procedures for some reason, then you need to directly contact us. Don't just ship things through the mail without getting particular instructions from Joe. Okay, so I do want to just that's chlorophyll A. That's the process of both collecting and filtering the samples and getting them into us for analysis. We have a little extra time though, and so what I like to do at the end of this presentation is to talk a little bit about your data. I'm kind of the data guy on the team. I'm the one who, when everything's in at the end of the year, I'll be making these reports for you. And then that usually takes a while. And then the report will come to you hopefully at the end of February or the beginning of March the next year. So I just wanted to kind of go through that report and tell you what you can expect to see. And I just completely randomly grabbed one of the currently existing ones, and it turned out it was Devil's Lake in Lenawee County. So you'll see the site ID on it. You'll get that latitude, longitude point. And remember, these reports are going to be made publicly available online. And it's not just you or your lake association that will be using them, but anyone who's interested in your lake, this is an easy way for them to get compiled data. They also can get the raw data off the data exchange, but this is just a really quick snapshot of all of your data. So it's really convenient. And I know that eagle biologists in particular are going here all the time and looking at the data that CLMP volunteers are collecting. This is what your primary data page will look like. And what we have is the whole page is divided into 6, and we have a spot for each of those main parameters. So there's a spot for the Secchi and the chlorophyll and the phosphorus and the dissolved oxygen. And in each of those, you'll have your current year's results as well as like a recent window, the last 5-year results, and then your entire history. And then for each of those, you'll see your whole history graphed out over time. The dissolved oxygen is a little different because I can't show an entire series worth of information for dissolved oxygen. What we see here is a particular day of someone sampling. It's one profile. And what I will often do for dissolved oxygen is look at all the profiles that you've created over the course of that year and pick the one that shows that point at which the stratification happens for your lake, when where you have this break where the dissolved oxygen is, and then suddenly it disappears. And most lakes have some point in the summer where that happens, especially if you're a mesotrophic or eutrophic lake. And then finally, we'll have a summary. And here's where we talk about some changes that are maybe happening and how that affects your trophic status. So that continuum of oligotrophic to hypereutrophic. That's the main page, but then there will also be all these other components. You will attach to the back, you'll get full-size graphs for each of those. You'll get, we're in the dissolved oxygen temperature parameter, you'll get your full every graph, every profile that you made. You'll get your score of the shore results, you'll get your aquatic plant results, all that will be attached into your report. And it's certainly the case that I make mistakes. And if you find a mistake, please let me know, and I promise to fix it fast. So just be in touch, and I'm happy to get things done quickly. So here's kind of, speaking of chlorophyll, here's just a close-up of like what one of those parts will look like. And you can see that we have these different windows. We have the current year, we have the past 5 years, and we have your whole history. Then we also give a little comparison of all the lakes that have registered that particular year. So you get a sense of, how does my lake compare to other lakes? Of course, this is just lakes involved in our program. It's not a comparison against you versus the entire state. So it is people who have opted in to the program. So it's a little biased in that regard. We don't have information on every lake in the state, so we just get you everything we can. Then over here on the far right, you'll see the Carlson TSI. That's the Trophic Status, Trophic State Index number. And this is a number that we use to determine where you fall on that oligotrophic to eutrophic scale. So let's just take a look at that a sec in a little bit more detail. We can come back to that. So what is this Trophic Status Index? Actually, I'm going to go one more, and I'm going to come back again. I'm just going to rearrange the order of these. And I like to think of it as a thermometer. So, and Eric mentioned this also, it's how we classify lakes based on their nutrients really is a continuum. You can go from low to high and everywhere in between. So it's not like you just fall into a particular category and that's you. You know, you might be right on the border between oligotrophic and mesotrophic, and that's just the state of your lake. Now, one of the pages of the reports that you will get will have this information summarized, and it will show this, you know, nutrient thermometer that you can look at, and you can see where your lake lands. Devil's Lake got a 36 for the number for summer phosphorus, and a 38 for chlorophyll, and so we're going to average those together. So altogether, average, this lake is a 37, which means it's right on that border between oligotrophic and mesotrophic. You'll notice they didn't have their Secchi disc data in here, and that's because they didn't do 8 measurements over the course of the summer. They actually only got 5 measurements in. So if you don't get 8 measurements in, then you don't get rewarded with this kind of end result of the number that tells your trophic status. So let's just back up one step here, and what these numbers actually mean. So the problem that we have is that we're trying to compare Secchi to phosphorus to chlorophyll. Now, all those are measurements of different things, as we've talked about, and they're all measured on slightly different scales. You know, Secchi depth is measured on feet, and summer phosphorus and chlorophyll, they're measured on parts per billion. So they're trying to measure different things, and you're going to get different numbers. What these equations that you see in front of you, what they do is they're basically converting all those numbers so that they can go on the same scale. And then you'll end up with a number that ranges for a normal lake somewhere between like a 25 and up to about 70 at the worst. So it's a way to put all of those 3 parameters kind of in a comparison so that you can look at them all at the same time. Devil's Lake got a 37 in 2019. Then you can see how in this last 5 years, it also had a 37 average. But then over the course of its whole history, it used to have a 40. So Devil's Lake is a lake in which the long-term trends are showing us maybe just a slight decrease in nutrients over time. It's kind of it's marginal. That may be year-to-year variation in there. For some lakes, it's more dramatic, and you might see a decrease of 50 going down, which would be eutrophic, and it might go down to 40 over the course of 20 years, where it's definitely in that mesotrophic category. So that's just an interesting way to think about how your lake is changing over time. Across the state for 2019, this is the results for the trophic status. So in 2019, which was the last year we did the sampling for CLMP, we had 264 lakes in total that got at least one of those trophic status numbers to us. And we have the slight majority of them are mesotrophic. And then we have about 100 lakes oligotrophic, about 30 eutrophic, and one lake is hypereutrophic, meaning it's at that really high nutrient level that's starting to even get a little dangerous in terms of possible oxygen depletion and fish kills. You know, we don't like to assign values to what is a good lake and what is a bad lake, but it is the case that the higher up on the nutrient level you go, the more dangerous it is for your fish community, for example, because there's a chance that you could have algae blooms, which then upon decomposition consume oxygen and can result in fish kills. So really the value of your lake is how you use your lake, and these numbers are just to give you a sense of where you fall on this scale, but we try not to assign values to the oligotrophic, mesotrophic, eutrophic. In general, long-term trends across the state, I did the analysis a few years ago, and at least with the CLMP lakes, which I have access to the data, I have noticed over time nutrient levels very slowly going down across the state of Michigan. So where we once had a lot more mesotrophic lakes, we are now starting to see more and more oligotrophic lakes, and actually a lot of these borderline, like right in between mesotrophic, oligotrophic lakes. A lot of lakes fall in that category that join the CLMP. So in terms of overall water quality, that is generally considered good, at least in terms of they're moving further away from that fish kill zone, right? The lower nutrients means you're less likely to have those algae blooms, which people generally do not want to have on their lake. In particular, if you go back in the 1970s and 1980s, you'll see a lot of lakes across Michigan have very high nutrient levels, but a lot of them, most of them have gone down considerably as things like septic systems get taken out and sewers get put in. That makes a huge difference to keep that nutrient level low. Zebra mussels, you know, not necessarily something that you want in your lake, but that has had a big impact on lowering nutrient levels in lakes. But I think more and more people are just aware of how bad fertilizers can be for lakes. So I think over time, we are going to continue to see a drop in nutrient levels. But over time, what we're seeing is more and more people love their lakes, maybe a little too much, right? So while nutrient concerns may be going down, what we are seeing is that impacts on shoreline habitats are just growing so much. Exotic plant spread is raging out of control, right? So nutrient threats may be declining a little bit, but there's new threats coming in for lakes as people are honestly just enjoying their lakes, maybe a little too much. You know, it's got to be done in a careful way so that the lake can maintain its integrity. And that's why in the CLMP we have added these other parameters over time. You know, we now have a way that you can analyze your lakeshore habitat, and we have these exotic plant watches, so you can watch for those incoming plants. So we're kind of pivoting the program to concentrate on those new threats, as well as not forgetting about the nutrients. And I think that sums it up for what I have. So we have time for questions. I think we said we're going to go 11:45. So with that, Joe can come back on.
Sure. And we did have a couple of questions come in while you were talking. And of course, if folks have other questions, they can ask them in the chat right now. The first question that came in close to the beginning of your presentation, Paul, was, I am new to this, but I believe our lake has been registered for quite some time. Are we able to do chlorophyll this year?
This might be one that I ask the other people to chime in because I can't quite recall.
Sure, Paul. I wanted to give you the first stab at it. Yeah, in general, if you're a new volunteer, even if others on your lake have been monitoring in the program for a while, and especially if you're working alongside them, new volunteers can do chlorophyll, even though this is an advanced parameter. If your lake and you are new to this program, then this is one that we'll have you hold off until next year to do. Paul, here's a good question for you. And you covered it, and I think the question may have come in before your slide came up, but maybe you could reiterate. Earlier, it was said that some lakes are clear enough to see the Secchi disc all the way to the bottom. If that's true, how do I double that number?
You don't double the number. In that case, you just go all the way to the bottom with your sampler, and you mark that depth on your data sheet. And yeah, that's all there is to it. You just go as deep as you can, even if your Secchi doesn't permit it according to the, those strict rules. So.
All right, thanks, Paul. Another question that came in: if a lake is being treated with chemicals for invasive plants, does that affect the timing of testing our various parameters?
No, you always stick to our timing. And if you get strange results because of treatment going on in your lake, then those strange results are just part of your data record. I mean, that is what is happening on your lake. So you always want to stick to our schedules.
That's great advice, Paul. And another thing I'll mention along those lines is that there is a place on your data forms where you can note any unusual conditions. So if anything unusual is occurring on your lake at the time you take that sample, you can put that on there so you remember. You may say there was an algae bloom visibly happening, or there was a lake treatment that occurred 2 days before. You can make that note. The only time, and we haven't gotten to this yet today, but the only time you might want to think about how chemical treatment for aquatic plants may impact your sampling is if you're out there sampling plants. We're going to go over the Exotic Aquatic Plant Watch here a little later this afternoon. And we do suggest that you avoid any plant surveys right after work has been done to kill plants, right? Because that will skew your results and you won't find the plants that were treated and potentially destroyed by that treatment. Another person, actually, Paul, if you wouldn't mind, on your slides, they wanted to see that trophic status thermometer slide again. Yeah, could you maybe talk about that again a little bit?
I love this slide, so I would love to talk about it because I think it's the coolest. So you often hear lakes called, oh, I have an oligotrophic lake. Oh, I have a eutrophic lake, and so on. But you have to remember that any category is really just a way to simplify it in our brain, but it's a spectrum. You can fall anywhere on here. And that's why in the reports, I'll often tell you, like, when you're right on that borderline between the two categories, because it's an understanding that life is a continuum. What else to say about this? If your numbers are wildly varying, like let's say you get 50 in your Secchi, but your summer phosphorus and your chlorophyll maybe are closer to 40 or lower. When I make these reports, I'll draw all those lines on so you can see how that spread. If for that particular example I just said, it may indicate that there's something else going on in your lake besides just nutrients. Maybe you have a high sediment load, and that is making you have a worse Secchi measurement as compared to like a phosphorus or a chlorophyll measurement. If that kind of thing isn't going on, generally the three numbers will all fall pretty close to each other. And usually when you have one strange outlier, it means that something else is happening here that you'd have to think about why that is.
Thanks, Paul. Had another question come in, and I can handle this one because, again, it's about when you can sign up for different things. And the question is from someone who says, I've been doing Secchi and total phosphorus monitoring on my lake for 10 years, but not through the CLMP. Do I still have to wait till next year to start chlorophyll monitoring in the CLMP? In that case, if you're an experienced person who's had other experience outside the CLMP doing lake monitoring, and you'd like to start off doing more than just the basic parameters in your first year with us, we encourage you to reach out to us and just ask. Let us know what your experience and your history has been, and we often will allow that for experienced folks that have some science background who have been doing lake monitoring before this. Another question that came in, why doesn't CLMP include nitrogen as a parameter? And I can start to address that too, and maybe my colleagues will want to chime in. As Eric talked about when he presented on phosphorus, for most of our lakes in Michigan, nitrogen and phosphorus are both obviously required nutrients for plant growth in our lakes, but phosphorus is the one that tends to be the limiting nutrient. And what that means is that if you just get a tiny bit more phosphorus in a lake, you can see a huge jump in productivity. But the nitrogen is present in enough abundance that you don't need, it just isn't as sensitive to changes. And so we've limited our monitoring to phosphorus because that tends to be the one that is the best sign of changes in your lake for lakes in Michigan. All right, another question. In very clear, shallow lakes, I believe the chlorophyll sample is not supposed to be taken at the very bottom for fear of touching the bottom, but 2 or maybe 3 feet off the bottom.
Yep. Yeah, don't hit the bottom with your sampling equipment. It would definitely, then you'd have to sit there for 20 minutes and wait for it to all settle down. Right?
Exactly. Yep. You mentioned that before, Paul, that it's important to avoid bumping the bottom with our sampling gear. Another question that came in, the person asked again, they said, in case I might have missed it, but can we start entering data such as Secchi measurements? And as I mentioned before, we are doing a lot of updates to the MiCorps website. And so if you try to enter data right now, you may run into a not available error message as we're doing some work on the database. If you do have old credentials for entering data from past participation, you may be able to do that now. But as I said, be patient. You might run into some times where it's not available to you. But once that is open and available for everyone and we're not doing any more work on the system, we'll be sure to announce that and let everyone know. Another question, where and when can we pick up our lake testing supplies and equipment? Because of COVID, we're not doing any pickup this year. We are mailing everything. And so once you enroll on the MiCorps website and choose your parameters, we will get that equipment for whatever you signed up for mailed to you directly, whatever address you give us as the lead volunteer that's enrolling, that's where that will be shipped. And Jean Roth at MLSA will be handling that like she has for years. The only exception for that will be dissolved oxygen and temperature monitors. Those meters, we have to hand deliver those. And so we're going to be working with partners throughout the state to get those out to the folks that sign up and are planning to borrow the meters that we own for this program. All right, that's the last question I see coming in. So Paul, unless you had anything else to say?
I look forward to seeing what you guys come up with this summer. I like looking at your data at the end of the year. just meet those deadlines, please, so that enables me to get working on it faster. And I can get results to you faster if you get that late October deadline for data entry.
All right, thanks so much, Paul. And before we break for lunch, there was one other question that snuck in. We'll stop questions after this. And I want to mention it because I've gotten a couple of direct messages about it too. Some people's lakes have a lot of different coves and bays and, aren't a perfect circle. In fact, most lakes aren't. And there's been some questions about whether it makes sense to sample in all of your coves and bays or just in one spot. In almost every case, it's better to just stick to one central location for sampling. But talk to us. If you're not sure, please contact us. You can reach out to any member of our team, or just, we even have a general email address. It's MiCorps@msu.edu. And you can reach out and ask your questions like that. Because in some lakes, there may be a clear distinction where they really function as two separate lakes. And in those cases, it may make sense to collect data from each basin. But in most cases, the protocols weren't designed for that, and you won't learn a whole lot more about your lake by doing all that extra work and paying all that extra money. So we'll talk to you about it, and we'll get that straightened out on an individual basis.